Hello friends, welcome to my channel Sam Biology Class. Today we will discuss about chemotaxonomy. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. Chemotaxonomy deals with the classification and identification of organisms, particularly plants, based on the differences and similarities in their biochemical composition. It is also called chemosystematics. It is also called chemosystematics. Once again repeating, chemotaxonomy deals with the classification and identification of organisms, particularly plants, based on the differences and similarities in their biochemical composition. Chemotaxonomy is also called chemosystematics. It relies on analyzing various compounds, mainly secondary metabolites produced by different organisms to establish evolutionary relationship and to classify organisms. Secondary metabolites are not involved in the fundamental metabolic pathways. However, they play a crucial role in plant survival by defending against biotic stresses such as pest and pathogen, and also abiotic stresses like UV radiation. Chemotaxonomy provides valuable data. Chemotaxonomy provides valuable data for distinguishing closely related species and varieties, thereby making it a powerful tool in plant identification and classification, especially when traditional methods are not sufficient. Both primary and secondary metabolites are studied for chemotaxonomic purposes. The phenolics, terpenoids, alkaloids and non-protein amino acids are the four important groups of compounds that are widely exploited for chemotaxonomic studies. These compounds exhibit wide variation in their chemical diversity, structure as well as function. Chemotaxonomy has been useful in the study of most popular plant families such as Malvaceae, Ranunculaceae, Polygonaceae, Magnoliaceae and Solanaceae. Chemotaxonomy is based on the principle that the chemical composition of an organism varies between species, thereby offering unique taxonomic characters. Now the question is how chemotaxonomy works. How chemotaxonomy works. works. There are three main steps in chemotaxonomic studies. First step is chemical analysis. First step is chemical analysis. Second step, comparison and classification. Second step, comparison and classification. And last step is determination of phylogenetic relationship. In first step, the chemical composition of an organism is analyzed, identifying the presence of various compounds like flavonoids, alkaloids, fatty acids and terpenoids. In second step, the presence or absence of these compounds and their pattern is compared among different organisms. Based on this comparison, we can determine that whether the organisms are similar or different in their biochemical composition. If two organisms have similar biochemical composition, we can say that these two are taxonomically related. If not, they will be taxonomically distinct. Based on these differences and similarities, scientists infer the evolutionary relationship and classify organisms into different groups. Applications. Chemotaxonomy has several applications. First one is plant taxonomy. Chemotaxonomic studies are widely used for the identification and classification of plants, particularly for distinguishing closely related species and varieties. In addition, it is also used for microbial identification and classification. Finally, chemotaxonomic analysis of different plants help us to find out the presence of new bioactive compounds, thereby helping in the discovery of new drugs. Friends, this is all about chemotaxonomy and its applications. For more updates, subscribe my YouTube channel. Goodbye and good night.